Dear students, now I am going to talk about gene regulation, positive and negative gene regulation. Genetic information on DNA as a series of genes. So, gene expression is how cell decodes this genetic information to synthesize the proteins required for cellular function. You studied all those processes in my last classes. So, RNA and protein synthesized in organisms during development or during maintenance or in response to the environment are responsible for organism characteristics. So, there are thousands of genes expressed in a cell determine what the cell can do. So, it is a normal process. You will find every cell expresses certain genes, not expressing certain genes, expressing certain genes at certain period. So, there is thousands of genes expressed in a cell determine what that cell is doing. So, how do cells know what types of proteins they need to manufacture? So, flow of genetic information from DNA to RNA then to the protein provides the cell several checkpoints for self-regulating its functions by adjusting the amount and the type of protein it manufactures. So, there are so many regulations or checkpoints are there. So, it is during the time of transcription it can be controlled, or else after the post transcriptional regulation, or else post translational regulation. So many, in this way what the gene express, what the gene going to be expressed will be regulated. So, factors that influence gene expression include nutrients, temperature, light, toxins, metals, chemicals and signals from other cells. So, all these you will find the temperature also. Just a small example, when you go to the sun you will be immediately tanned. So, when you are not expose yourself to the sun, you are not so tanned, sun tanned. So, similarly the metabolic pathway depends on our so this is how the gene expression is affected by the factors such as nutrients, temperature, light, toxins, metals, chemicals, signals from the other cells. So, gene regulation is the process of controlling the genes in a cell to be expressed to make protein. So, it is what type of proteins should be manufactured, or else this is nothing but gene expression. Different cells in a multicellular organism may express different sets of genes even though they contain the same DNA. The set of genes expressed in cell determines its uniqueness. So, these are the embryonic cells, blastomeres, cells of blastula. All these have same genetic composition because they are all derived from the zygote by cleavage. You are studying the developmental biology. So, how these cells, these are the stem cells, they become muscle cell, blood cell, nerve cell, then cardiac cells, liver cells, intestinal cells, all these, So, they cell differentiation, that is nothing but gene expression, turning certain genes on and certain genes off. So, this is what express or repress, that is going to make the differences in the gene expression and from the stem cells to derive into muscle cell or nerve cell. This combination of turning genes on and off is what dictates the function of cells in the organisms. So, genetic regulation or gene regulation varies slightly between prokaryotes and eukaryotes. The type of regulation varies in prokaryotes and eukaryotes. Eukaryotes is the complexity, so that's why. So, the genes are regulated primarily at the level of transcription and also in the level of translation level. So, gene regulation in prokaryotes The DNA of prokaryotes is organized in a circular chromosome. You know, you have studied how exactly last semester. Proteins that are needed for a specific functions are encoded in the units called operons. So they are in the functional unit encoded, that is called operon. So gene regulation is adapted to provide maximum growth in an environment, and gene expression controlled mainly at transcriptional level in case of prokaryotes. So bacteria regulates the gene expression so that only gene product or proteins needed are produced. So for example, if the bacteria is grown in a medium containing the glucose is the energy source, then you can see the enzymes needed for the glucose metabolism is produced. Similarly, if it is in the lactose medium, the enzymes required for lactose metabolism will be produced. This is how the gene expression, the gene products or proteins needed are produced. This is achieved turning on certain genes. Synthesis of mRNA induced only when those gene products are needed. So when the gene products are not needed, turning off the gene and the synthesis of mRNA is stopped. So this is what you can see, the mRNA synthesized. So if these proteins are required, if these proteins are not required, this mRNA is not synthesized. That is what you can see, turning on and turning off genes. So there are three types of regulatory molecules that can affect the expression of the operon. So the operon will be expressed or repressed or not expressed depending on the three types of regulatory molecules they are called. So they decide whether to operon to be expressed or that proteins are synthesized or not to synthesized. Those are called repressors. These are the type of proteins that suppress the transcription of a gene in response to an external stimulus. So in other words, the repressor keeps gene off. So repressor here, when it is sits on the operator, so if the RNA polymerase sits on the promoter, it cannot able to slip or slide and to express or to make mRNA because the repressor prevents. So this is called repressor. So it shuts the gene off. Then another opposite to this, activator. So activator is a type of protein, and the transcription of a gene in response to an external stimulus is it is turning on the gene. It sits on here so that you can see the RNA polymerase can bind to the promoter region, so that mRNA synthesis is initiated. So transcription is on, so turning the gene on. Then another substance called inducers. These are small molecules that either activate or repress the transcription depending on the needs of the cell. So inducer may act as a repressor or as an activator depending on the requirement. So those are called inducer. So in prokaryotes, the type of regulation, there is a positive regulation and negative regulation. So positive regulation means gene expression is quantitatively increased by the presence of specific regulatory element called as positive regulation. So in this gene expression, the gene product is quantitatively increased. So this is called positive regulation. So the binding of an activator or effector, so to DNA, activates the transcription to begin. So DNA bound activators can regulate the transcription by helping the binding of the RNA polymerase. So this is positive regulation. Activator going to bind and it brings the transcription. So it enhances the transcription. If it is not going to bind, no transcription. So this is called positive regulation. So what exactly negative regulation? Opposite to that. So gene expression is quantitatively decreased by the presence of specific regulatory element. This is called negative regulation. So here the regulatory element is called repressor. So the binding of the repressor inhibits the transcription. So the gene product is quantitatively decreased. DNA bound repressor prevents the RNA polymerases from binding to the promoter region, so that the no transcription. So in the presence of a inducer substrate required to allow the transcription, if in the presence of inducer, so repressor cannot bind, so that transcription will takes place. So this is called negative regulation. So positive and negative regulations are not exclusive. So you should not think only positive for a operon, negative for, not like that. In some system, in some operons, positive and negative regulated utilizing two regulators to respond to the different conditions. Both the operons are positively regulated or negatively regulated. So it is regulated by two regulators. So it's in some operons. So in catabolic pathway, you can see it is regulated both positively and negatively. So it is lactose metabolism. So where you can see it is a catabolic pathway. So it is regulated positively and negatively. In biosynthetic anabolic pathway, in the production of the a protein or production of you can see the polymer, then you can see it is negatively regulated. The amount of that gene product will inhibit the further production of the the mRNA. So that is called the negative regulation. So all this gene regulation in prokaryotes extensively studied and proposed for the first time by Francis Jacob and Jacques Monod. So it is called the Jacob and Monod model. They proposed operon. The concept operon is proposed by this is Monod and this is Jacob. So this is what you can see, the repressor control of lac operon transcription, which was based on genetic experiments they did on with the E. coli, Escherichia coli, and proposed a simplest negative regulation in prokaryotes. So they are awarded with Nobel Prize in 1965 for this model or the operon concept. Promoter, that is, you know what is promoter, that controls the expression of the lac operon, is regulated by two different proteins. One of the proteins prevent the RNA polymerase from transcribing, it is called negative control. The other enhances the binding of the RNA polymerase, that is activator, positive control. So negative regulation of the lac operon. So it is a bacterial regulatory system for lactose utilization or lactose metabolism. So lac operon regulated by repression, specifically the product of the lac-I gene. There is the lac-I gene which is upstream to the promoter of the lac operon. Okay. So this gene will be expressed, this system, whenever there is a lactose is absent. When the lactose is absent in the medium, this protein inhibits the transcription of lac operon because these enzymes are not required. So ZYA, it is these genes are they are responsible for the three enzymes, that is beta galactosidase, permease and transacetylase. All these are required for lactose metabolism. These genes are inhibited when the lactose is not in the medium. So it is regulated by the gene that is called the gene product lac-I. It inhibits the transcription of lac operon. It is called lac repressor. So lac repressor is a protein that is coded by this lac-I gene. The mRNA produced by this produces a protein called repressor. This repressor protein going to bind the operator region, so that the RNA polymerase which binds on the promoter region cannot open the open complex or else open the DNA, because it cannot, it is inhibited by the repressor. So lac-I is constitutively expressing gene constitutively expressing gene is, there is no regulation for this gene, lac I. So it is continuously it is producing repressor protein. So when the lactose is absent, this repressor is bind to the operator, not allowing to express these genes which are required for lactose metabolism. In the absence of lactose, lactose is an inducer. The repressor blocks the expression of the lac operon by binding to the operator, which is downstream to the promoter and upstream to the transcriptional initiation site. So this is a transcriptional initiation site. So the repressor bind to the operator so that transcription is turned off. The operator consists of a specific nucleotide sequence that is recognized by the repressor, which binds very tightly, physically blocking the initiation of transcription or else the movement of the RNA polymerase, so that it cannot open the DNA. So no transcription. The lac repressor has a high affinity for lactose. Small amount of lactose in the medium, lac repressor bind and causing the dissociation. When the lactose is present, what happens? You can see lactose will bind with this repressor protein so that it get dissociate from the operator region. It cannot no more bind with the operator, so that the operon is turned on. So all these enzymes are produced. So they bring the lactose metabolism. So this lactose inducer, the repressor cannot remain bound to the operator. Transcription of the structural genes can proceed. So this is what you can see. So the lactose inducer. So in the presence of lactose, so the repressor lactose complex going to form. Repressor lactose complex. So which binds to the RNA, now RNA polymerase binds to the promoter site and gene is transcribed. In the absence of lactose, repressor protein binds to the operator site, O site. So the lac operon block the RNA polymerase from reaching the P site, so that you can see the RNA polymerase cannot able to open the complex. So that is transcription complex. When the structural genes are translated, allows the once the structural genes are expressed, so these enzymes are synthesized, it allows the greater flux of lactose molecules which are metabolized by beta galactosidase, convert the lactose into glucose and galactose. So that the energy will be supplied. When all the lactose is metabolized, then the lactose dissociate from the repressor, so it is no more bind with the repressor. So that what happens, you can see once again repressor going to bind on the operator and stops the because there is no lactose in the medium, so the enzymes are blocked now or mRNA not going to synthesize it. And how exactly this is about negative regulation. Now the positive regulation. So the regulatory mechanism in E. coli allows catabolite repression or the reduced repression of the genes when growth in the presence of the glucose. So you see, it is in the presence of the glucose in the medium. What happens? Let us see. This is called positive regulation. Glucose is very easily metabolized, readily energy giving molecule. You know that. So it is preferred when in the presence of lactose and glucose, the bacteria will prefer the glucose. So it is a preferred fuel molecule over the lactose. Hence the expression of lac operon prevented when the glucose is present. The strength of the promoter is determined by its ability to bind RNA polymerase and to form a open complex. So the promoter, you can see it is the strength of the promoter depends on the binding of the RNA polymerase. So the promoter of the lac operon is weak, and consequently the lac operon is poorly transcribed upon the induction. Whenever there is a glucose, you can see the lac operon is poorly expressed. So because glucose is preferred, there is a binding site upstream of the promoter for a protein called catabolite activator protein, CAP it is called, catabolite activator protein. So this is upstream to the promoter region. This is upstream means this in the right side. When the cap protein binds to, then it distorts the DNA so that RNA polymerase can bind easily transcription of the lac operon. So this cap will bind this cap site when there is no glucose, only lactose. Okay, so low amount of the, you can see the when the glucose is in the medium, so cap cannot going to bind, because so cyclic AMP attaches to the cap and allow it into the bind to the DNA. So when the glucose is in the low amount, so when the glucose is low amount or else in the absent, so the cap can able to bind to the cap site. So because along with the cyclic AMP, that is another messenger. So in order to bind the cap must first bind cyclic AMP. So a second messenger synthesized from the ATP by the enzyme adenyl cyclase. You can see, so the ATP adenyl cyclase enzyme. So you will find the formation of the cyclic AMP when in the presence of cyclic AMP. So there is, you can see the cap, the catabolite act it is the catabolite activator protein cap. So it can able to bind to the cap site. Okay, in the presence of glucose, circulating cyclic AMP levels will be low when there is a glucose is present, so that cap cannot able to bind. As glucose level decreases, the concentration of cyclic AMP increases, activating cap, which binds the cap site, stimulating the transcription. So when the glucose is present, the high amount of then there is no cyclic AMP is formed. Now the cap will not bind the DNA without cyclic AMP. So transcription will occur in very less amount. So this is how exactly the in the presence of glucose, in the absence of glucose, or very low amount of glucose. Very low amount of glucose, cap site is bound by the cap, so that the lactose operon is operated, or this lactose is also utilized, because low amount of glucose. When the glucose is more, so that you can see the cap cannot able to bind, so that the low transcription. So this is what you can see, the gene regulation. This is called positive regulation. So this is about prokaryotic gene regulation.